I would like to move to our first presentation by Hansjörg Lehnherr, who will talk to us about basic characteristics of phages. This presentation will be, Hansjörg, you may already try to share your presentation.
I'm on it.
Yes, and I will just explain that this presentation will be quite long. It will take around 40 minutes, but please bear with us because Hansjörg will be covering really the most important aspects and characteristics of phages. And it is important to understand this in order to be able to follow also the future sessions. So Hansjörg, please, the floor is yours. And if you have any questions to Hansjörg, please type them in the chat box. I will collect them and present to Hansjörg after his presentation.
Yes, thank you, Margareta, for the introduction. I'm today going to introduce you to some very basic
We can't see your presentation, sorry.
You can't see my...
No. Can we? No.
Still not?
I don't see it.
I try again.
I can see it.
You can see it?
We can see the presentation.
Okay, sorry, then please. Oh, now it's coming to, yeah. There was a problem with, okay, great.
Yes, now it's working?
Yes.
Okay, where was I? Basic characteristic of bacteriophages. Yes. But first I would like to give a quick detour to explain the motivation why we do work with bacteriophages. Last year, the World Health Organization published a priority pathogen list. And that was really, really scary. Infectious diseases are back, and the causes of these diseases are antibiotic resistance. And that is very bad news for our high-tech medicine. Routine surgical procedures like an appendix can become life-threatening if the patient catches a hospital-acquired infection that can no longer be cured with the standard antibiotic we use. And patients that are immunocompromised for a liver transplantation, there the situation is much, much worse. And the outlook is very bleak. The situation with AMR will not improve if we do not find valid alternatives. And so this report is really a plea for help that we should do more research to find alternatives to fight AMR. On this slide, you see 6 pathogens which are on the top of the World Health Organization list. And some of them, like they cause tuberculosis or typhoid, I'm only aware of them out of textbooks or in historical novels. I didn't realize that they present again big problems in our medicine. Bacteria are really very amazing creatures. They can react very, very quickly to selective pressure. They have a very short generation time, produce a lot of progeny, and they have a high mutation rate. And that makes them very adaptable. And they have various ways how to become resistant against an antibiotic. Just 2 examples, an enzyme called beta-lactamase cleaves the antibiotic penicillin and thereby destroys it, and the bacterium that expresses this enzyme is resistant. And a lot of our medicine is based on penicillins or its derivatives, 1st, 2nd, 3rd, and 4th generation. And the bacteria are adapted, and they are now able to cleave almost any of these 2nd or 3rd or 4th generation penicillins. Or another example, bacteria have an efflux pump where they can efficiently pump out an antibiotic like tetracycline before the tetracycline can work inside the cell. Once such a resistant bacterium is present, it will not disappear again. And under selective condition, such resistance mechanism will spread among the microbiological community. The more antibiotics we use and the more selective pressure we build up in our hospitals or anywhere else, the faster this development will go. And if we look at that from a One Health perspective, we are really living in a very interconnected world. Then it doesn't really matter where the selection occurs, if it's in human medicine or in veterinary medicine or in animal husbandry. If the bacteria are resistant, they will spread throughout. So finger pointing, you are guilty to do this and you are guilty to that, that doesn't really help. We are all in the same boat. We have to address this question jointly. We have to try to reduce the amount of antibiotic we use. So we have less selective pressure and a slower development of antibiotic resistance. And we really have to find alternatives to fight pathogenic bacteria. And better these alternatives also work when the bacteria are already AMR. Now we at PhageEU and many others believe that phages are one way to do that. With phages, we can really target attack pathogenic bacteria. And the mechanisms by which phages work are very different from the mechanisms antibiotics work. And so phages still work when a bacterium is AMR. When we talk about phages, we from the audience very frequently get questions. That can be basic one, what is a bacteriophage? Or more tricky ones, they're viruses, are they somewhat dangerous, or can they mutate and become dangerous? And today I'm really going to try to present you with some very basic concept on what a bacteriophage is. Let me start with these pictures. A bacteriophage is something very, very, very small. We don't see it by the naked eye. In the light microscope, they're not visible. Only in the electron microscope, we start to see these futuristic particles. We have a head and a tail that can be long or sometimes short. And we can see here tail fibers, very filigrane structures. And if we see something in the electron microscope, the question is, how big is it really? And I have two more pictures. On the left, we see a bacterium that is soon going to divide. And these tiny white dots, these are the bacteriophages. And on the left-hand side, we have a picture where the bacteria are stained blue. And the bacteriophages are these tiny green, yellowish dots. So a bacterium is approximately 2 micrometers in size, and the bacteriophage is about 0.1 micrometer in size, so 100 nanometers. And now it's clear something here is off. My bacteriophage is way too big compared to the bacterium. So my bacteriophages are not drawn to scale, but if I would paint them to scale, we just see a dot, so you wouldn't see anything. When I make a schematic diagram of a bacteriophage, it's really just a protein structure that carries a little bit of genetic material in its head. And in this form, a bacteriophage is not a living organism. Biologists define a living organism, something that can react to external stimuli, that has its own metabolism and is able to generate progenies that at one stage or they don't look like the parents. A bacteriophage does not have a metabolism. So the bacteriophage doesn't see anything, doesn't smell anything, doesn't swim around. It simply does nothing. There is some energy stored in a bacteriophage in the structure. And movement we see from bacteriophage is simply Brownian motion in liquid. A bacteriophage needs a bacterium to come to life. And that's how I like to explain it, what happens. if a bacteriophage finds a bacterium, and it has to be specific for it, then it can attach on the surface of the bacterial cell. And that triggers the injection of the genetic material into the bacterial cell. And now this bacterial cell is being reprogrammed. It usually has a program out of one bacterial cell make 2, out of 2 make 4, out of 4 make 8. That is exponential growth. And the bacteriophage, or rather the genetic material of the bacteriophage, tells the cell, stop that, rather use all the energy to produce new bacteriophages. Eventually, the bacteriophages accumulate within the cell. They feel confined, and so they start from inside to destroy the bacterial cell wall. And this cell wall bursts open due to the internal pressure, and the bacteriophages are released in the environment. And they literally have nothing else in their head than to repeat this process. Now, for the bacterium, that is very bad news, because the bacterium dies if it bursts open. And this killing capacity is really what we base the bacteriophage applications on. Now, this cycle we call a lytic growth cycle. It's very, very important. It's really the basis of everything we're doing. And if I describe it like this, it sounds very easy, but it's far from easy. Bacteria do not like to be reprogrammed. They can defend themselves. But for the moment, I will just stick to this lytic growth cycle. And during my talk, I will always come back to it. Now, one question we can address, where do we find bacteriophages? As they're so small, we really have to look carefully, but it turns out we find them everywhere. In soil, in water, in air, in all the living creatures. And we have an idea just how many of them are. And to do that, I want to look at a special habitat, namely the ocean. Every milliliter of seawater contains approximately 1 million bacteria and about 10 million bacteriophages. And this is kind of a natural equilibrium. But this equilibrium is not static, so it means nothing happens, but it's highly dynamic. And the driving force is the living organism, the bacterium. The bacteria will start to grow, and so we see an increase in their cell concentration. And with a certain delay, the bacteriophages find their host and start to amplify in them. So the concentration of bacteriophages increases. And as they eat more and more of the bacteria, the number of bacterial cells will drop. And then the bacteriophages do not find a bacterial host anymore. And even so, they do not have an energy turnover. They're not immortal. They can be inactivated by UV light, or they can be eaten by krill. So the bacteriophage concentration goes down again. The bacteria come up, the phages come up, and so on. This natural equilibrium is actually very, very important, because it keeps the nutrient cycles in the oceans running, the carbon cycle, the nitrogen cycle, and so on. Bacteria are released or opened up, lysed open, and their nutrients they had inside are released into the environment and can then be used either by other bacteria or by higher organisms. And now I have a question. I know you can't answer, but I ask it anyway. How many milliliters of seawater do we have on Earth? And as nobody probably has an answer, I make a quick calculation. The surface of the Earth is about 500 million square kilometers. 70% of that is covered by ocean, and the average ocean bed is 3.5 kilometers. So we calculate that, we sum 10 to the 9 cubic kilometers, or we turn that into cubic centimeters. And we have, I told you before, seawater contains approximately 10 to the 7 bacteriophages per milliliter. And we come to an astounding figure of 10 to the 31 bacteriophages in the ocean. Now, if you compare that 10 to the 31 bacteriophages, approximately, if we calculate 10 to the 29 bacteria, but only 10 to the 9 humans. So we are clearly in the minority here. And the question is, how do we really know that they're out there? And that's science, metagenomics. We take a sample and we study all the genetic material that is in there, and we can quantitate that, and that gives us an insight into the composition and diversity of our environment. I have a second slide, and that is the human gut. Every one of us in the digestive tract carries about 10 to the 15 bacteriophages and about 10 to the 14 bacteria. And that really is a safety argument. Bacteriophages inside do not do any harm to us. It actually turns out the more we learn about them, we realize that for a healthy gut microbiome, phages are very important. And if you have a disturbance there, if you have too few phages, then that can lead to disease. A question that always comes up, how do you work with the bacteriophages if you never see them? And of course, we could use an electron microscope to make pictures of them. But you see, this is a rather big machine. It fills almost an entire room. So not every researcher has access to an electron microscope. And all the others, they have to use classical microbiology to deal with bacteriophages. So in the laboratory, we grow bacteria on plates or in liquid, and then we try to grow the bacteriophages on these bacteria. And this is an example of what we're doing. Like we take an agar plate, we add a lot of bacteria to this plate, and just a few bacteriophages, we incubate the plate. And after overnight incubation, we see these holes. So the bacteria have been grown into a nice opaque lawn, and these holes, we call them plaques, are the result of one bacteriophage landing in a spot, finding a host, making this lytic growth cycle once. The progenies are released, they go through the lytic growth cycle. Their progenies are released, they go through the lytic growth cycle. And if that happens 6 or 7 or 8 times, so many bacteria are eaten from the lawn that we start to see this clearance zone. Now, this experiment is really old. Already the discoverers of bacteriophages observed such a phenomenon. Avery in 1915 called it glassy bacteria because you can now see through this bacterial lawn. And Darrell in 1917 already had the idea that something is eating the bacteria. And that's why he coined the term bacteriophages, bacteria eater, that we're still using. Now, such a method we can really use for a lot of things. For example, we can use it to count bacteriophages. If we have here a sample with an unknown phage concentration, we simply make a dilution row. Then we put these dilutions on a plate, so each dilution on one plate, incubate overnight, and then we take the sample where we can actually see the single plaques. We count them, and then we calculate. On this plate, I count 9 plaques, and the dilution rate I used to make this plate was 10 to the 8. So I can calculate that we have 9 times 10 to the 8 plaque forming units per milliliter, and that's the unit PFU per milliliter we use to describe the concentration of bacteriophages. What we can also do is we can isolate bacteriophages from nature. So if we have an environmental sample with an unknown phage concentration, we do the same. We add the bacteria, we add the same sample, and we check the next morning if we detect plaques. Now, this method or the isolation from nature really has some limitations. We need to be able to grow the bacteria. So they need to be able to make alone, otherwise we could never detect plaques. So uncultural bacteria, even so we know they're out there and we know they have bacteriophages, we will never find them. And also bacteriophages are very, very specific. So it really depends on the host, on the bacterium, what we find. If we have here a host of Escherichia coli, we will only find Escherichia coli bacteriophages. If the host is Staphylococcus aureus, we only find Staphylococcus aureus bacteriophages. And so the question is, why are bacteriophages so specific? Why is this the case? And going back to the lytic growth cycle, at the beginning, the bacteriophage has to recognize its host. It has to bind on the surface of the bacterium. And whatever is on the surface of the bacterium can be used as a receptor for the phage to bind. So there are phages who bind to flagella, there are phages who bind to fimbria, there are phages who bind to membrane pores, there are phages who bind to glycopolysaccharides on the surface or lipopolysaccharides. There are phages who bind to lipoteichoic acid and so on. So whatever is on the surface can be used as a receptor. And once the bacteriophage binds, this triggers the injection process. But it becomes quite clear. A bacteriophage that recognizes a membrane pore will not at the same time also be able to recognize a teichoic acid, or one that recognizes teichoic acid will not bind to a lipid. So this gives the specificity to the bacteria, and this also gives the specificity to the bacteriophages. And we find significant differences between the two groups of bacteria, like the gram-positive ones with this setup of cell wall, or the gram-negative ones with this setup of the cell wall. But also within the groups, like Listeria is gram-positive and Staphylococcus is also gram-positive, but they do not have the same extension, the same receptors on the surface of the bacterial cell. So to be able to attach and inject, the bacteriophage is very specific. And this entire thing we can actually extrapolate. The bacteriophages that bind to a gram-positive bacterium, they will also not find the receptor on a plant wall, cell wall, or on an animal cell membrane. The structure on the surface of the bacterium are simply not present on any eukaryotic cell. And so a bacteriophage will not be able to enter the cell of a higher organism. And now I hear a what-if question, like, oh, but pinocytosis, a bacteriophage is taken up into a eukaryotic cell. And in the lysosome, the structure is degraded, we have only the genome left, and that now manages to come into the nucleus of a eukaryotic cell. What happens then? And I have an answer for that. Nothing will happen, because the bacteriophages are highly specific for their bacterial host. Viruses are specific for the eukaryotic cells. And while these two kingdoms on Earth, they split a long time ago and do not really interact with each other other than when we have an infection. We see they have the same genetic codes and they have the same building blocks of life, but they are absolutely incompatible when it comes to DNA synthesis or protein synthesis. So this genome in the nucleus of a eukaryotic cell will not be expressed, no proteins will be made, no bacteriophage particle will be made, nothing will happen. And now I come to another point. Many people have problems with mutation rate. They see that as something highly dangerous, the organism mutate, and out comes a monster. So what if a bacteriophage mutates, they have a high mutation rate, what will happen? And to give a little bit of a feeling to that, I would like to make an example. If you have a chicken, that chicken lays an egg, and out of that egg hatches a chick. What is the probability that this chick will turn into an eagle? Now, this is something we can relate to, and we think that will never happen. However, if we look at that from an evolutionary perspective, this is actually something that is very realistic. Every chicken has all the genes that are necessary to fly. And nature can now come and make a few mutations. One mutation, the chicken will lose weight, become a smaller bird. And another small change, the flight muscles of this chicken will become stronger. And very quickly in evolutionary terms, we can turn a bird that cannot fly into a bird that can fly. And so the question now is, can we mutate the bacteriophage so it will end up as a coronavirus? And the answer is simply no, because no matter how we mutate this bacteriophage, no gene within this bacteriophage has anything to do with any gene in the coronavirus. And we can mutate and mutate and mutate, and it will never happen to be like that. This distinction between prokaryotes and eukaryotes is simply too big to overcome for a mutation, which is a point mutations on a small change in the DNA. My next point is a theoretical experiment. If I have here on the left-hand side one bacterium and on the right-hand side one bacteriophage, will this bacteriophage find its host? And the answer is no. There's no chance that this bacteriophage will find the host because it cannot swim. It is not targeted, it has no chemotaxis or whatever. It doesn't see that there's a bacterium over here. So it will move around here with Brownian motion. It is very happy on its own. Now, nature has found a way. And the nature solution is the following. The bacteria start to amplify, exponential growth, and eventually the bacterium will swim over here to the bacteriophage, and the phage will find the host and start a lytic growth cycle. And when that happens, we will re-establish the natural equilibrium we find in nature. But now we already know such a natural equilibrium we find in nature doesn't really prevent this bacterium from being pathogenic, from causing a disease, or it also doesn't prevent it from being antibiotic resistant. And so the question is, how are we going to deal with these bacteriophages? And the answer is very, very simple. We have to artificially increase the bacteriophage concentration. And this is an underestimate. We use 10 to the 6, 10 to the 7 bacteriophages to fight a handful of bacteria. And with such an artificial excess of bacteriophages, we manage to reach bacteria, even if only a few of them are around, and we can actually succeed in either reducing them significantly or even eliminate them. And that is really the basic when we talk about phage therapy, how we are going to apply bacteriophages. And of course, you can now immediately answer, oh, such a high concentration, 10 to the 7 bacteria, does that have any environmental impact? And our argument is no. Let's just take an example. We have a chicken gut with a chicken microbiome, and this chicken has Salmonella, indicated here as blue bacteria. So we feed the chicken 10 to the 7 bacteriophages specific or per milliliter specific against Salmonella. Then they come into the gut, they will find the Salmonella, they will grow on them and eliminate them. And for all the other bacteria in the microbiome, these Salmonella-specific bacteriophages will not do anything. They simply do not see them because they do not have the right receptor on the cell surface. We can do such a thing in a closed environment, like a chicken gut is a very confined environment. The moment these bacteriophages come out into the nature, if they come into the sewage treatment plant, there's such a huge dilution effect that they will be simply diluted out. And what we can artificially produce, the concentrations, they simply are not sufficient to disturb the natural equilibrium in nature. And so with the bacteriophages, we will never eliminate the bacteria from Earth, and we will not going to do any harm to the environment. Now here, the theoretical concepts I talked about, we see there are already some in place. We see applications in poultry or in aquaculture, in food, and several clinics are already successfully applying bacteriophages in human medicine. I'm not going to go into details here because you will hear a lot of that from our next speaker. And I'm coming to my last slide. There are some questions which I did not address today. For example, are all bacteriophages suitable for phage therapy? Or do all bacteriophages use the lytic cycle, or are there alternatives? Or how quickly do bacteria become resistant to bacteriophages? And how do we react to that when it happens? And of course, if I left you with more open questions than answers, then you're very welcome to ask these questions, not only in the short Q&A, but also later. And then I might address these questions at a later stage, at the second installment of the PhageEU Academy. And with that, I thank you for your attention, and I'm very happy to answer a few questions.
Indeed, Hansjörg, thank you very much. Yes, a lot of, I think, information to digest, especially for those for whom phages are a new concept. As we said, this is the first session of PhageEU Academy. There will be more, so we will be diving even deeper into the phage world. So all the questions you will have, all the questions you will leave with us, we will try to address them later. And actually, after our meeting, you will receive a satisfaction survey where you also will be able to indicate topics you would like us to cover in the future. I think your presentation, Hansjörg, was very smooth and understandable, as we don't have many questions, but we do have 2 questions. One very specific, I would say very complicated, even if to some people it seems simple. Another one, I think, a bit more straightforward. I will try to share them now with you, Hansjörg, and with the other participants, that you can see them, and you can maybe try to respond to it. But let me try to find it for you. Can you see it?
Yes.
Yeah. So the first one is, that you mentioned phages are generally considered to have narrow host range. Given the limitations in the isolation of natural phages that were highlighted, what is the possibility that we are simply missing entire groups of phages with a relatively broader host tropism?
That we are missing phages on nature, that's definitely the case, because there's so many out there that we might not always see them. But the problem with the narrow host range, we can solve. We use not a single bacteriophage in a treatment, but we compose cocktails with phages that have different receptors. And that's we are able to cover organisms which have a high diversity, like Salmonella or E. coli. We will never reach 100%, but that is not the goal. If we can cover 90% of an organism, we are very good on our way.
Right. I hope it answers the question. Otherwise, we may continue also exchanging on the chat during the next presentation. And the second question, it is simple to some. However, for me, it's too complicated even to repeat it. So please,
I'm trying to read it 1 second.
Take your time to read it. It's a lot of numbers, a lot of math. So take your time to read it and try to maybe address it.
The specific question, how it is asked, I can answer as no. If we choose the conditions that the bacteria are growing as a lawn, and we dilute the phages so that only a single phage manages to land on there. At that stage, we cannot overestimate the concentration. Because one bacteriophage cannot make 2 plugs. That is not possible.
Okay, let me check the chat. Okay, thank you for your talk. There is another issue, question. I was wondering how feasible you think it is to use phage therapy in plants, trees, to fight, for example, quarantine organisms. And why are the hurdles? What are the hurdles?
Okay, if we treat something on the surface of a leaf, then that's very, very, very simple. The moment you have to go inside the plant, like the bacteriophages have to be taken up via the root or injected in the phloem and the xylem to distribute within the plant, that's very, very tricky. But there are projects or phage groups who are work on plant application. So the feasibility is there. What was the second question?
Yes, and there is another one. Participant is asking that it has been found that there is a change in the classification in bacteriophages post-2021. Would pre-2021 phages have to be renamed, or would they still use the old names?
The nomenclature just changed, that we are no longer build our nomenclature on the structure, like on the structure we see in the electron microscope. So we had phages with a large tail and tail fibers that were called Myoviridae. We had some with flexure tails that were Syphoviridae. And we had the short one, I said, that was a Podoviridae. That was the old structure. And that was not considered to be good anymore, because the structure really doesn't tell you anything about the bacteriophage. It doesn't take into account on what host it grows, and the features of the bacteriophage are not determined by this structure. And so this old nomenclature was abolished to be replaced by a better one that is really has a species kind of concept. So we now have a genus and a family and yes, all the old phages, so all the T4s and lambdas and P1s, they have to be renamed. It doesn't mean that we can't say phage T4 anymore, but they now have a specific name.
Okay, there are more questions coming, and that's good because we still have some time. Hansjörg was very efficient. So Hansjörg, a very interesting question. Sorry, because it's so. How about bacteriophage resistance issue in comparison of antibiotic resistance? I think it's a topic that interests many people.
Okay, that's a topic that was way too big to be answered very shortly in this first session.
Maybe it's a good topic for the future to evolve.
I can give a very, very brief answer, then say for the illustration of the answer or for details to this answer, you have to come to the next session. But a resistance to an antibiotic for the bacterium is a gain of function. So as I said, the bacterium develops a new enzyme, or it gains a new efflux pump, or it becomes somehow adapted to the antibiotic. It doesn't take it up anymore. This is always a gain of function. The bacterium has a new property. And this property under selective condition is very, very beneficial. If you want to become resistant to a bacteriophage that is mechanistically different, that's always a loss of function. For example, you can simply throw off your flagella. Then a flagella-specific phage will no longer be able to infect you. So you're resistant against that phage. But you also no longer have a flagella. And so you no longer swim around, you no longer have chemotaxis. And that's a huge, huge disadvantage for a bacterial cell. There will be enormous counter-selection for maintaining the salmonella, the flagella, sorry. And so we will see in nature, phage-resistant bacteria, they pop up and are immediately selected out. Then they come up and are selected out, come up and are selected out. And so that makes a huge difference between antibiotics and bacteriophages. And that's why with the bacteriophages, we're not really risking to make the same mistakes or have the same problems as with the antibiotics. And that's one point in short why we believe that this will be very, very helpful to fight AMR.
There are indeed more questions about resistance, so maybe they will add some additional elements. There is a question, when a bacterium evolves resistance to an antibiotic, can it make the bacterium more susceptible to phage?
Yes. Yes, there are experiments that go on to see if we can see synergy between the antibiotic or synergy to and the phage. And we observed that if you become phage resistant, you become suddenly more susceptible to the antibiotic, and the other way around, that can also work. For example, just a slight idea, this efflux pump that pumps out tetracycline. If you imagine a phage that binds to this efflux pump, then the bacterium can internalize this efflux pump, then it is resistant to the bacteriophage, but it now no longer can pump out tetracycline, so it becomes sensitive to tetracycline again. several situations, that's just one example, but several of these examples have already been studied.
Okay, and there is also an interesting question. Is there any evidence that immune cells can attack phages? If yes, how and which cells?
Okay, are several groups, like in Poland is an important one, that study all the kind of immune reaction between phages and the immune system. So if you take a phage sample and you inject it in your blood, then your immune system will recognize them as foreign, it will react, it will make antibodies, it will stimulate the entire immune system. But the phages we have in our gut are a healthy immune system does not recognize them as foreign. And so from whatever we have in our gut, we do not see the immune system triggered. And phages are also rather small. They can go through the barrier between the intestine and the blood. They end up in the blood. And when that happens relatively frequently, but the concentrations that come in there are relatively low. And these concentrations under normal circumstances do not trigger our immune system. Our blood is usually filtered in the liver and in the spleen. And so phages, as being a protein particles, are simply filtered out and digested.
Okay, I think I will still address two last questions because we are now running a bit out of time, but please continue typing in your questions. As I said, we will try to address them in the future sessions. So there is a question that just came. What is the stability of phages at different temperatures, pH, and to disinfectants?
Okay, the last thing is difficult because I need to know which disinfectant it is. But very basically, as the bacteriophages do not have a metabolism, disinfections usually do not do anything. That's very, very general. Now, temperature, the phages have the optimum where the host has their optimum. So if we have a bacterium that likes 37 degrees in our intestine, the bacteriophage will also have 37 degrees as their optimum. And their protein structures, so if you start to cook them, they will denature and lose their biological activity. So anything above 60, 65 degrees is not good for a phage particle. Unless you select for it. Like if you have a hot spring, the bacteria are adapted to temperatures 80 degrees, 90 degrees, 100 degrees, then you'll find that their bacteriophages are also stable at these environmental conditions. pH is a little bit the same. If you use pH 14 or pH 1, of course, a bacteriophage particle is not stable, but in the normal range, bacteriophages are very stable.
Yeah. Okay. Okay, and the very last question I will ask to you, but then Hansjörg, you can also maybe take your time, go through the...
I will go through the chat and try to answer that.
See which questions haven't been addressed, because there was a follow-up question about plants, but I will just ask you the very last question, and the response probably is not short. Would it be possible to create a bacteriophage through engineering to broaden the spectrum of bacteria it can infect? If yes, is there already a way to do it?
Okay, we are able to manipulate bacteriophages. So to exchange a tail fiber, add there another tail fiber, that's absolutely possible. So we can already engineer bacteriophages to switch host. But there's a certain limit to that is not, the adsorption process is not the only limiting factor. If you have to take over the host, you need to use the RNA polymerase of the host, you need the DNA polymerase of the host, and the most important thing is you need to use the ribosome of the host. And if the ribosome doesn't recognize your mRNA as mRNA, you will not be expressed. And so that's how we make a nomenclature, how we type bacteria. If a bacterium has a different ribosome, we classify it as another type of bacterium. And so you can play a little bit like a bacteriophage that recognizes Salmonella. You can engineer it so it recognizes E. coli because they're very, very closely related bacteria. But you will never be able to engineer a bacteriophage against Salmonella to eat Listeria. That simply is not possible. You can try that genetically. You can give it a receptor for a Listerium phage, but then already the injection mechanism will not work because the bacteria are so different.
Perfect.
So yes, we can do a lot with CRISPR-Cas and all the mutant gene editing technologies. We can really do a lot, but it has its limitations and they're set by nature.
Great. Thank you very much, Hansjörg, for your presentation and for all your replies to the questions. And now we are moving to Jania Koziek, to another